Welcome back. This is our second lecture on Chapter 9, Muscles and Muscle Tissue. Today we're going to see at the microscopic level how muscles actually contract. We're going to put together that electrical activity, which is, remember, action potentials from Chapter 11, to contractile activity of the muscle. So this is putting those two pieces together. This is called excitation contraction coupling. And I thought this was an interesting picture before we start looking at the details of how this muscle activity actually occurs. What we're seeing here is a microscopic view of where neurons and muscle cells meet. These are called neuromuscular junctions, and this kind of dark color, this black or dark brown, this is the end of a motor neuron, a neuron that controls muscle cells. And at the end of each, here are our axon terminals, and they are sitting on the surface of muscle cells. This is called a neuromuscular junction. So here's kind of a cartoonized version of what we just saw. This is a neuromuscular junction. Sometimes you will also hear it called a motor end plate. But reality is, this is just another synapse. A synapse exactly like what we learned in Chapter 11, except instead of one neuron talking to another neuron, here we have a neuron in yellow talking to a muscle cell in pink. So this is just another type of synapse. And what we're going to look at here is how does this neuron pass an action potential to a muscle cell? The good news is it does it exactly the same way that a neuron passed an action potential to another neuron at our previous synapse. So let's look first at the neuron. Action potentials traveling down the neuron, which we already learned in Chapter 11. When that action potential gets to the axon terminal, there are vesicles inside that contain the neurotransmitter acetylcholine, the same one used in our example in Chapter 11. So when the action potential reaches the axon terminal, voltage-gated calcium channels open, calcium comes in and binds to the vesicles. The vesicles then travel to the edge of the cell, dumping the acetylcholine into the synaptic cleft, exactly like we saw in Chapter 11. The acetylcholine diffuses across the synapse, ultimately binding to chemically gated or ligand gated channels. These channels open, allowing the ions to pass through. So sodium rushes in, maybe a little bit of potassium goes out also, but more importantly, sodium is coming in, depolarizing the muscle cell. And all along the membrane of the muscle cell, the sarcolemma, the exact same proteins are present as were present along the axon that we saw in Chapter 11. We have voltage-gated sodium channels, voltage-gated potassium channels. We still have our leaky channels. We still have our sodium-potassium pump. So all of the things involved in generating an action potential are present. So we get an action potential, and that action potential is now going to pass along the surface of this muscle cell. And here's what that action potential looks like in graph form. Very similar to an action potential in a neuron, but there are a few differences. Remember, all cells in your body are slightly negative inside compared to the outside when at rest. In a neuron, we saw that it was negative 70 millivolts at rest, but in a muscle cell, still negative, but it's negative 90 millivolts at rest. So at rest, negative 90, During depolarization, the exact same events happen. Sodium voltage-gated channels open, we depolarize, we get to positive 30, voltage-gated sodium channels close, voltage-gated potassium channels open, so we repolarize back to negative 90. However, we see that there is no hyperpolarization in muscle cells. So the big difference is here, negative 90 millivolts at rest and no hyperpolarization. Other than that, all of the events are exactly the same in skeletal muscle as we saw in an axon of a neuron. So back to our neuromuscular junction. Now we're zoomed out a little bit so that you can see more of the surrounding area. Here our action potential travels down the axon until we get to the axon terminal. At the axon terminal, the events that we just saw occur. We generate an action potential in the muscle cell, and that action potential begins to spread along the sarcolemma. Well, the sarcolemma, remember, has those infoldings where parts of that cell membrane fold in. We call that a T-tubule or a transverse tubule. And since that T-tubule is part of the sarcolemma, it's made of the same stuff, as this action potential spreads along the surface of the muscle cell, it also travels down the T-tubules. And here we're just zoomed in on that area. So here we see the action potential spreading along the surface of the sarcolemma. We also see it now traveling down the T-tubule. Think back to our previous lecture. Remember, a T-tubule on each side of it had a part of that sarcoplasmic reticulum that was kind of swollen. That was our terminal cisternae. Two of the terminal cisternae, one T-tubule was called a triad. So here we see a triad. Action potential traveling down the T-tubule. Well, where the triads are found, the terminal cisternae are anchored to the T-tubule with special proteins called ryanodine receptors. Ryanodine receptors are also similar to a voltage-gated channel. And when the action potential passes along the T-tubule where a ryanodine receptor is found, it causes that ryanodine receptor to change shape. First, let's think about what was the job of the sarcoplasmic reticulum. Well, what was the job of a smooth endoplasmic reticulum? One of those jobs was to contain calcium. Any calcium that's found inside of a cell, we have said, is stored away inside of an endoplasmic reticulum because calcium likes to attach to stuff. And in general, we don't want calcium to do that. So if it's inside of a cell, it's stored in a smooth endoplasmic reticulum, and the sarcoplasmic reticulum is smooth endoplasmic reticulum. So the terminal cisternae is full of calcium. But when this ryanodine receptor changes shape, there's actually now a pathway from the inside of the terminal cisternae to the rest of the inside of the muscle cell. What's actually happened here is this action potential has caused the release of calcium. Action potential arrives, ryanodine receptors change shape, calcium exits the terminal cisternae into the interior of the muscle cell. Let's go back to something that we talked about in the previous lecture. Remember, the actin has those small little dimples, the active sites, and that's where myosin heads really want to attach. But when a muscle is at rest, those active sites on actin are actually covered up by tropomyosin. Tropomyosin is lying on top of those active sites, blocking them. And that tropomyosin is held in place by troponin. And now let's also think about what we keep saying about calcium. Calcium likes to attach to stuff, so it's normally kept stored away in the smooth endoplasmic reticulum or the sarcoplasmic reticulum. But now calcium has been released into the interior of the muscle cell, so it's going to start to attach to stuff. And here, what it's going to attach to is troponin. When calcium is present inside of a muscle cell, it binds to troponin. And since troponin is a protein, anytime you attach something to it or take something away from it, we change its properties, we change its shape. We attach calcium to troponin, troponin changes shape and pulls the tropomyosin out of the way. So now what we see is those active sites on actin are exposed, and the myosin head is going to attach to those active sites, forming a cross bridge. So let's put everything from the past couple of slides together. The action potential arrives, causing calcium to enter the interior of the muscle cell, which then allowed for cross bridge to form. So the action potential resulted ultimately in cross bridge formation. Up to this point, everything we've talked about is excitation, contraction, coupling. And I feel like this is one of those things that just hearing it explained one time usually isn't sufficient. It's much better if you can see this in action. And there are videos, I know there are videos on YouTube. There are also videos on our study area in Mastering A&P. So if you go there, if you go to YouTube, you can Google or you can look up excitation, contraction, coupling. If you look up excitation, contraction, coupling, Pearson, it will bring up your textbook's video of this. There is a more current video of this, I believe, in the study area that can kind of show you animated all of the events that we've talked about. Also coming up, we're going to see what happens because of this cross bridge formation and how this results in contraction. So when we get through that, I will tell you about another video that you can watch that shows the rest of the story. And one final thing that I really want to point out, I think your book kind of drops the ball a little bit on this diagram. Before we go to the next slide, I want to point out that at rest, when the cross bridge has not yet formed, when the myosin head is in this position, even though we don't see it, there is a molecule of ADP and a molecule of inorganic phosphate, both attached to the myosin head. So we don't see it, but really there would be a molecule of ADP kind of attached right here, and a molecule of inorganic phosphate attached kind of right there. And that's just sitting there, which will play an important role in the next bit of information. Let's look now at what happens as a result of that cross bridge formation. So everything that we just talked about has happened, and now we have our cross bridge. Here's our ADP and our inorganic phosphate that we just talked about. Once a cross bridge forms, now it's going to start to get really important with that whole every time you add something to or take something away from a protein, it changes the protein. That's going to be what drives this entire bit that we're going to talk about now. We attach the myosin to the actin, so that changes the protein. That causes the inorganic phosphate to fall off, which strengthens the bond between myosin and actin. It makes that cross bridge stronger. When that bond forms stronger, that knocks off the ADP, adenosine diphosphate, and when the ADP falls off, that changes the shape of myosin so that the myosin kind of pivots forward. This is called the power stroke. During this time, we are pushing the thin filaments towards the H zone, towards the M line, and the same thing is happening on the other end of the sarcomere from the other direction. Your book kind of puts both of those steps together into one thing, but in reality, first the inorganic phosphate falls off, strengthening the cross bridge, then the ADP falls off, causing the power stroke. Once we have had the power stroke and the myosin head is bent forward, ATP comes along, adenosine triphosphate. The ATP attaches to the myosin head, pulling it apart. It breaks the cross bridge. Once the cross bridge is broken, that splits the ATP into ADP and inorganic phosphate. The ADP and inorganic phosphate has broken, which causes the myosin head to pivot back into what we call the high energy position or the cocked position. Right back exactly where it was on our previous slide prior to forming the cross bridge. That's where that ADP and inorganic phosphate came from. Now, once this happens, as long as there are still action potentials still coming from that neuron, then there would still be calcium in the cell and the actin active sites would still be exposed. So the myosin head would do it all over again, and we would go through this cycle several times. Going through that cycle one time does not noticeably shorten the muscle. It actually takes many of these cycles to really fully contract a muscle. So it actually takes many action potentials to fully contract a muscle. But this whole thing that we just looked at is called the cross bridge cycle. And just like with our excitation contraction coupling, you can find lots of videos of this that instead of just hearing it explained and looking at pictures, it actually shows it in video format step by step. But your muscles, once they've contracted, they have to relax again. So how does relaxation occur? What happens when a muscle is done contracting? Well, there's no more stimulation from the motor neurons. We stop sending action potentials to the muscle cells. And once we stop sending those action potentials, the ryanodine receptors close and calcium stops leaving the sarcoplasmic reticulum. Also along the sarcoplasmic reticulum is another protein called calsequestrin, which pumps calcium back into the sarcoplasmic reticulum. So calcium levels inside the muscle cell very quickly drop, which means calcium is no longer attached to troponin. Troponin moves back to its original shape, pulling tropomyosin back over the active sites on actin. Myosin heads can no longer attach to actin. So everything is back to its original shape. And remember the protein titin, the little springs inside the sarcomere? They pull everything in the sarcomere back to its resting length. Let's end this lecture on something that can be a little bit creepy. Rigor mortis, all right? Rigor mortis is contraction of muscles after death. And what we see here is a corpse experiencing rigor mortis. What's actually happening here is the muscles along the back are contracting, raising this corpse up. So what is rigor mortis? What causes it? Think back to that cross bridge cycle. Every bit of that was the result of chemical reactions, and chemical reactions don't require life. Chemical reactions occur even in the absence of life. Following death, there is no stimulation from motor neurons. All action potentials stop. All muscles completely relax. There is no tone, which we've seen in lab now. There's no muscle tone. There's no tonus. Muscles go completely limp. And it stays like that for a while until eventually calcium begins to leak into muscle cells. Either they leak in from outside of the cells or they begin to leak out of the sarcoplasmic reticulum. But regardless of where this calcium comes from, now there's calcium inside of your cells. And calcium likes to attach to stuff. Doesn't take life to do that. That's a chemical reaction. Calcium attaches to the troponin. Troponin changes shape, pulling tropomyosin out of the way. And now the active actin sites are exposed. Myosin binds to actin, cross-bridge formation. We go through several cycles of the cross-bridge, and that contracts your muscle. None of that takes life. That's just chemical reactions. But what does take life is the formation of ATP. Inside your muscle cells, there's a big store of ATP. And ultimately, we're going to deplete that ATP as we go through the cross-bridge cycle. And once that ATP is depleted, well, now we don't have anything to break the cross-bridge. So the muscles contract, and we eventually get to a point where we've contracted the muscle, there's a cross-bridge formed, but there's no ATP to break that cross-bridge, so the muscles stay contracted, and that's rigor mortis. Given enough time, even the proteins like the actin and the myosin begin to break down. And once they begin to break down, then the muscles go back to a relaxed state. They can't stay contracted because the proteins themselves are denatured. And that's when rigor mortis completes. That's when rigor mortis finishes. And a lot of times on TV shows, you'll see that someone can come along and look at a body in rigor mortis and give you an idea of how long this person has been dead. The way that works is you can look to see, has rigor mortis begun? Is rigor mortis still going? Or has rigor mortis completed? And you can kind of factor in things like, what's the environment like? How hot or cold is it? Is this person in water? Is it humid? Is it really dry? Things like that affect how long rigor mortis takes. So I can't really tell you this is how long it takes for each of those steps, because it's completely dependent on the environment. But you can, if you are trained in this, see rigor mortis and some other things that happen after death and get kind of a gauge for how long somebody has been dead.